Hi guys, here you go. We are starting today respiration. Okay, the class is split in 4 videos. So the first one we are going to talk about the citric acid cycle. We can also say Krebs cycle, which is the name of the guy that has discovered this cycle, a German. And also TCA cycle, which is 3-carboxylic acid cycle. Okay, I prefer to use TCA. Probably you are going to hear more TCA, but it's the correct way. The chapter of the book is the 16 about the TCA cycle and the 19 about oxidative phosphorylation. So these are the two topics that we are going to study now. So this first video class is just an introduction about respiration. Okay. We have already discussed about glycolysis, which is the first step of respiration. Okay. So we are not going to detail this. We have already studied. We have here the TCA cycle, which is the step 2, and also the step 3, which is the oxidative phosphorylation, which contains the respiratory electron transfer chain or transport chain. Okay, so as you can see here, every step is connected to each other by this electron production. So it means that glycolysis and the TCA cycle are providing substrate to the oxidative phosphorylation step. Okay, in the form of NADH, FeADH2, and also succinate. So glycolysis, as we remember, it starts with the glucose degradation to pyruvate. But then pyruvate is degraded until acetyl-CoA by this pyruvate dehydrogenase complex. We are going to study this enzyme specifically in the last class. But here, not only glycolysis feed the TCA cycle. For example, if you are not intaking a carbohydrate into your body is going to start to degrade fatty acids or proteins, for example. So proteins release amino acids, and then these two kind of compounds can be converted into to acetyl-CoA. Okay, so acetyl-CoA is substrate for many pathways, not only the TCA cycle. Okay, so here is just showing the transformation of acetyl-CoA plus oxaloacetate into citrate. So citrate is a combination of two compounds, contains 6 carbons, and then the citrate keeps the cycle running. And remember that respiration is the opposite of photosynthesis in terms of gas exchange, because here we have release of CO2 here, and also here, and we have the consumption of oxygen. Remember, photosynthesis is the opposite. It's assimilating CO2 and producing oxygen. So the oxygen production from photosynthesis is basically the base of Earth life. Okay, so every animal respires, and for respiration they need oxygen. Remember what I asked you: what is important, what is essential to our life? You may have many things important, but essential, some of the things are oxygen. Okay, so without oxygen, there is no animal life. This comes mostly from photosynthesis. So the TCA cycle is providing electrons in the form of NADH and FeADH2, which is going to be used to produce electrons in the oxidative phosphorylation step. So this is used to create a proton gradient, as we saw in photosynthesis. And then we have ATP synthesis. So ATP synthase in the mitochondria is very similar to the ATP synthase of the chloroplast. Okay, so they are quite similar in terms of structure, in terms of similarity between the sequence of amino acid sequence, and the mechanism of ATP synthesis is basically the same. You have a gradient of protons, and the ATP synthesis is dissipating this gradient, and then you have ATP synthesis. Okay. So these steps are connected to each other, as you can see here, but they are physically separated. So glycolysis occurs in the cytosol, and TCA cycle oxidative phosphorylation in the mitochondria. But you are going to see in the last class that we have some metabolite channels that connect all of these steps. We are going to see this. They are separated physically, but they are very the regulation is unique. Like the respiration has some points of regulation, glycolysis, TCA cycle, and also oxidative phosphorylation. TCA cycle occurs in the matrix of mitochondria and the oxidative phosphorylation in the inner membrane. Okay, so both two of these phases in the mitochondria, one in cytosol. So glycolysis, it's important to generate ATP and ADH and also FADH2, which is going to be used not only in the respiration but many other reactions that need, for example, ATP and ADH. But this is very important for ATP production in the mitochondria. The TCA cycle is the same and has one production of GTP, which can also be converted to ATP. So mostly of the enzymes, not mostly, but many of these enzymes here can produce NADH or FADH2. So this is going to be used in the last step to produce ATP. So remember that plants has chloroplasts, and chloroplasts can also synthesize ATP. Actually, plants in the light, chloroplast is the main site of ATP synthesis. It means that plants can not make a respiration, but the respiration is lower in the light. So plants respire more in the dark. Okay, but animals don't have chloroplast, right? So the majority of the ATP generated comes from mitochondrial respiration. One thing that has to be clear, and we are going to see this in detail also in the next class about fatty acids and amino acids, is that TCA cycle is not only providing substrate to the oxidative phosphorylation. So TCA cycle is a kind of hub in a network. We are talking about network. The TCA cycle can, for example, produce substrate for fatty acids and sterol synthesis and many amino acids. For example, alpha-acetylglutarate is substrate for these amino acids, oxaloacetate is substrate for these amino acids, and these amino acids also. So for example, if this cycle is running in the mitochondria, the citrate can leave the mitochondria that is transported that can pump citrate out of the mitochondria, and this, for example, these fatty acids in plants, they can the synthesis involves chloroplast, and also the cytosol, sometimes the endoplasmic reticulum. So there are many transporters that can bring something in or pump something out of the mitochondria. So TCA cycle is really connected to many other pathways besides the oxidative phosphorylation. So put everything together, we are going to have to see something complex like that. Don't be scared. Here we have the system of transport of electrons of the mitochondria, the complex 1, complex 2, 3, 4, and here you have the ATP synthase. Repair that the structure is more or less the same of the ATP synthase of chloroplast, and the mechanism is very similar. What is the difference between photosynthesis and respiration? Here we don't need light, so the electrons that you are seeing here are created by different proteins that can degrade, for example, the NADH. Remember the NADH that has been produced in the glycolysis and also TCA cycle. So this is consumed by the complex 1. So the complex 1 converts NADH into NAD, and then you have electrons being carried by different proteins. The complex 2 degrades succinate into pyruvate, and here you have also electrons. So you see that many components can provide electrons to the pool of ubiquinone. In the photosynthesis, we saw something about plastoquinone carrying electrons to one side to the other side. Here is ubiquinone, it's very similar also. And look here, you have the complex 1 and complex 3 and complex 4 have protons being pumped from one side to the other side of the membrane. So here you have the matrix of the mitochondria, this is inside of the mitochondria, this is the inner membrane of the mitochondria, and this is the intermitochondrial space, intermembrane space, okay, inside of the mitochondria also. So these protons are pumping from the matrix to this intermembrane space, and then the ATP synthase dissipates these protons from here to here, and then you have the synthesis of ATP. It's very similar to the chloroplast, okay? Uh, the main difference is because this is not necessary to have light. Plants can respire, for example, you can also respire in the light or in the dark, okay? Um, and plants has one alternative pathway of respiration, which involves this AOX, the name of alternative oxidation. Um, this protein can take some electrons. We are going to see that this is very important for plants to tolerate stress. Okay, so you see here that you have many DH enzymes, so IVDH, HDH, GDH, IDH, MDH, PDH. So they are all dehydrogenases, and most of them can produce NADH or NADPH. Okay, so these are very important compounds to be degraded here. So you see here NADPH can also be degraded here to produce electrons. So these electrons can be provided by many components of this mitochondrial proteome. Okay, so this is a very brief introduction. In the next video class we are going to focus on the TCA cycle. Okay, bye bye.